Okay, so now you have that sequence, and so what is the architecture that you're going to follow to solve this problem?
Right. So what we've done, there are a couple of available architectures, and what we do in this paper is actually combining two architectures and making an ensembling of those two already powerful architectures, and we show that it gets even more powerful. So the first one is going to be based on language models. So the top part, where you give a sequence and you feed that to a language model, and it's really BERT for proteins. A BERT trained on protein where each amino acid would be a word, and the whole protein would be a sentence, and the BERT has just learned how to reconstruct the sentence or the protein with the missing amino acids. In BERT, you also have the next sentence prediction. They don't do this. In general, people who train proteins, BERT for proteins don't do this because you don't have a protein that follows another protein.
Yeah.
I think there might be ways of using next sentence prediction for other tasks that involves proteins, but overall in the community, we'll see people just focusing on the masked amino acid task.
So the way that it was trained, it'd be like, let's say you have 20,000 different amino acids. Oh no, sorry, 20,000 different, amino acids, right? It could
amino acids, yeah. I think we can, in average, the length is in the 500, 500.
So let's say you have 500 amino acids. It will mask the first one and then try to predict it on the basis of the other 499, and then just kind of sequentially mask more and more in the same way that you would do it for language. Okay. And these are all just like transformer based. They're very similar to, yeah.
Correct. Yeah, it's literally a BERT.
Yeah. Okay. And who produces these? Are all open source?
These are almost. So there are some open source, some less open source. I can cite a few. So the one we're mainly using in the paper is ESM, that is produced by Facebook. I think it's evolutionary scale something. Yeah.
Scale modeling. Yeah, that one.
Yeah, modeling. So ESM is very nice because it gives you a lot of different models that are quite updated quite often. So that's and basically if you follow the code here, you can see that you get from a given sequence, you can end up with a representation. So here it's for structure prediction. They went quite far, and it was really nice to follow this GitHub for as time went, because now it's like a lot of layers on top of the previous knowledge. And
you're using the embeddings though.
For example, I think things you can do like this. Yeah, so embeddings is one of the things we're using for representations at Shiru, and in this paper, that's the one we're using.
Okay, cool. So basically, you feed in the sequence, and you get a bunch of representation. You get this sort of like embedding. So this is the language model representation, and okay, I cut you off. So this was the first approach. First approach is to just go the language model route.
Right. And here you could train, ahead of the model or a couple of last layers. So you can freeze this whole transformer encoder layers and just fine tune basically the last bits for a downstream task, like here the subcellular localization. And frankly, you would have already a very good result. This worked very well. Yeah.
Okay, so that's that's one approach. You guys then combine
Right.
this in an ensemble. So
Right.
what's the other one? Yeah.
Right. So the idea was to when we thought about this paper, AlphaFold just came out, and we were like, okay, for sure. So we were giving the sequence to the model and giving the information about the structure. For sure, will make it better, right? Because as we said, I mean, depending on the downstream task, but here we're benchmarking for subcellular localization. We believe that giving not only the sequence, but the overall, I mean, the information about the structure will really help them all, making any type of inferences. So what we did is we used the output of AlphaFold, which are PDB files.
What does it look like? What is this PDB file?
So PDB file it's canonically what is used for representing 3D structure. Yeah, 3D. So it's a little bit confusing because PDB is the name of the database and also the name of the file. But yeah, it's a set of coordinates where you would have each atom in the molecule. Each atom of each amino acid, and you see it means carbon, but there is also Ca, which is the carbon alpha, which is kind of the center of the amino acid. And so AlphaFold really gives you every single atom. But if you want to focus on kind of the overall shape, you can just take the carbon alphas, the chain of carbon alphas, and it will tell you overall, where each amino acid end up.
Oh, that's cool. Almost like the center of mass.
Yeah. It's where it's what makes the difference between the backbone and the side chain. And the side chain it's another layer of complexity. Is one of the reasons it was so hard to fold proteins in silico. Because so the whole backbone can fold, but also the side chain can move around and impact the folding. So
okay, so you feed it into AlphaFold, and basically what you get is something like this, and now you're going to convert this into some form of graph.
Correct. Yes. So from here, we decided to build a graph where each node is basically the carbon alpha I was talking about. So basically representing the amino acid, and there is an edge if two carbon alpha are close enough, and that will give you a graph of overall structure of the protein.
Okay, so a couple of questions. Why, if they're close enough, put an edge as opposed to always add an edge and add a weight that corresponds to the distance?
Yeah, that's a good question. So, well, if you want to use a graph-based approach, you probably don't want your graph to be fully connected for either computational reasons, and also learning patterns. If you do some pooling, for example, so some graph pooling, you want the graph to have a topology. You don't want everything to be connected and be solely relying on the weight of the edge. I mean, technically you could, but then you might just want your program to be learning at which weight you kind of discard an edge to extract the topology. So
it sounds like a heavier learning task. You might need much more data and hyperparameter optimization.
Right. It's one of the things where here you can pick a threshold that is informed by experts, where we know that two amino acids are actually chemically connected if they are below a given threshold. So we can use those thresholds and this knowledge.
Yeah, that's cool. And then Is this also a similar reason for why you decided to only focus on the carbon alpha as opposed to like just the entire base? Okay.
Yeah, basically baby steps. We try the first thing, we see if it works, and then if it works well, we kind of move forward. Making training a graph that represents every single atom might come with some other computational challenges for sure.
Yeah. Okay. So you feed it all into this graph. Well, first you represent it as a graph, and then you feed it into some graph neural network, and you get some form of like, these are like similar as like embeddings or something.
Right. Yeah. Then you can extract the representation as well.
Got it. Okay. And I think I see here, I mean, this maybe for people who are just tuning in for audio, the idea is that you then sort of concatenate both representations.
Yeah, it's just a fancy way of with trial and error, we ended up with this ensembling architecture. You could have just focused on the mean or train, concatenate and train the final layer. And with a couple of iteration, we ended up finding this kind of random architecture that worked the best.